We will begin our journey of human development at ovulation. Ovulation releases the secondary oocyte, which is picked up by the fimbriae and taken into the fallopian tube, where it waits for the sperm. Once the sperm arrive, they head towards the egg to both sides of the fallopian tube. When the sperm reaches the egg, it must penetrate several barriers in order to enter the egg. And these barriers are the corona radiata, which are a circle of cells around the secondary oocyte, protective layer, and then we have a clear layer called zona pellucida, and then we have the cell membrane. In order to penetrate these barriers, the sperm releases enzymes called acrosomal enzymes that are in the acrosome portion of the sperm. These enzymes help to digest these three barriers, the corona radiata, the zona pellucida, and the plasma membrane, in order to enter the egg. So we see just one sperm entering. There are many other sperms trying to enter the egg at the same time. How does the egg stop the other sperms from penetrating and bringing more genetic material into the egg than is needed? We only need one haploid set of chromosomes to enter the egg. The egg accomplishes this by carrying out what is called the fast sperm block and a slow sperm block. So let's see how that happens. As soon as the sperm penetrates the egg, sodium channels open and sodium rushes in, thereby depolarizing the egg cell membrane. This depolarization stops other sperm from entering. This is called the fast block, where the egg immediately blocks further entry of sperm into the egg. Next, we have the slow block. When the egg cell becomes depolarized, calcium channels open and calcium rushes into the egg. We know that calcium can cause exocytosis. The egg contains cortical granules that are arranged in a circle. When the calcium enters, these cortical granules undergo exocytosis and release a fluid that surrounds the egg cell membrane, causing a physical barrier for the other sperm. We call this physical barrier the fertilization membrane, and this is the process of the slow block against polyspermy, because we don't want more than one sperm and one set of haploid chromosomes to enter the egg. Once the sperm penetrates the egg, it induces the secondary oocyte to complete its meiotic division. So the oocyte undergoes meiosis II and becomes a mature ovum. So now we have a haploid ovum with its set of chromosomes and a haploid sperm with its set of chromosomes. Together, these two haploid sets can come together and make a diploid zygote. We call these the pronuclei, the male and female pronuclei that merge together and form the zygote, which is the first diploid cell of the embryo. This zygote now divides. It divides into two cells, and then the two cells divide again and make four cells, and so on, until a cluster of cells are formed called the morula. These stages of division, which are mitotic divisions, is called cleavage. The final structure that is formed is called the blastocyst, and this blastocyst leaves the fallopian tube and implants into the endometrium of the uterus. So here is implantation. Here's a closer look at the blastocyst. We have the blue-colored cells forming the trophoblast and the pink cells forming the embryoblast. The trophoblast becomes the placenta and the embryoblast becomes the embryo. Once the implantation occurs, the cells of the trophoblast release a hormone called the human chorionic gonadotropin, or HCG for short. HCG forms the basis of the pregnancy test. HCG Its function is to act on the corpus luteum and maintain it. The corpus luteum, thus maintained, continues to produce progesterone. Progesterone maintains the endometrial layer. So let's go over these steps one more time. So once the embryo implants, it releases HCG. HCG then activates the corpus luteum and maintains it. Corpus luteum releases progesterone. Progesterone maintains the endometrium, and once the endometrium is maintained, there is no menstrual period, and the woman is officially pregnant. So how are twins formed? We have two types of twins: identical twins that are genetically identical, and fraternal twins who are more like siblings. With identical twins, you start with a single egg and a single sperm. Once fertilization is complete, a zygote is formed. This diploid zygote then goes on to divide. After the first cleavage division, the cells, instead of remaining together, they separate. And each separated cell then becomes its own embryo, which is how we get two identical offspring. With the fraternal twins, we start with two eggs. The woman produces two eggs during ovulation, and therefore two different sperm fertilize these two eggs, and we get two zygotes. And these zygotes are quite different from each other, like two different siblings, and we end up with two non-identical offspring. Once the blastocyst has implanted, the cells of the blastocyst continue to divide and grow, but they also become differentiated. So at a very early stage, the fate of these cells is established. So we have three germ layers that are first established. We have ectodermal cells in blue, mesoderm in red, and endoderm in beige. Now, continuous with the ectoderm is the amnion. The amnion surrounds the amniotic cavity, and continuous with the endoderm, we have the yolk sac and the allantois. The trophoblasts, in the meantime, continue to multiply and form what is called the chorion. The chorion is the placenta. It is the embryonic portion of the placenta. Now let's take a look at the fate of the three germ layers: ectoderm, mesoderm, and endoderm. The ectoderm becomes the skin and the nervous system. So the nervous system actually develops from the skin cells. The mesoderm becomes the muscular system, the cardiovascular system, and the skeletal system. The endoderm lines the digestive and respiratory tracts. The amnion and the amniotic cavity contain the amniotic fluid. And the function of the amnion is protection, and it provides movement for the growing embryo. The protection is from changes in temperature that occur outside the embryo, and also acts as a cushion against external trauma. It acts like an airbag. The chorion forms the future of the placenta. The allantois is the umbilical cord, and it forms the urinary bladder in the future. The yolk sac provides nourishment for the growing embryo. It forms the early blood cells, and it also forms the early germ cells. Now, these germ cells will differentiate into male or female gametes, which is the sperm or the egg, but at first they form in the yolk sac. Before we study fetal circulation, let's review the pathway of blood through the heart. Deoxygenated blood from all parts of the body enters the right atrium via the vena cava, and from there blood flows to the right ventricle. The right ventricle then pumps the blood to the pulmonary trunk, which takes it to the lungs via the pulmonary arteries. In the lungs, the blood is oxygenated. Oxygenated blood then returns to the heart via the pulmonary veins, where it enters the left atrium. From the left atrium, the blood flows into the left ventricle, which pumps the blood to the aorta, which takes the blood to all parts of the body. The fetal circulation, on the other hand, is quite different because the lungs are not functional in the fetus. The fetus does not breathe while it is in the uterus. The function of the lungs is taken over by the placenta. The placenta provides the fetus with oxygenated blood, which enters the heart via the umbilical vein. The umbilical vein makes a detour around the liver. Normally, blood will flow through the liver, so majority of the blood makes a detour around the liver, and this modification of the umbilical vein is called ductus venosus. That is the first modification of the fetal circulation. This makes sense because the fetus does not feed while it is in the uterus, and so the liver is not functional. So the blood that takes a detour around the liver enters the heart and enters the right atrium. From the right atrium, the blood goes directly into the left atrium through an opening called the foramen ovale. The foramen means opening and ovale means oval. This opening closes around the time of birth. This movement of blood from the right directly to the left atrium makes sense because the blood does not have to go through the pulmonary trunk and into the lungs that are not functional. So the foramen ovale is the second major modification of the fetal circulation. The foramen ovale closes around the time of birth. The rest of the blood that is not flowing directly into the left atrium does go through the pulmonary trunk, and again there is another shunt along the way. The blood can also be shunted directly into the aorta to avoid sending it to the lungs. This shunt is called the ductus arteriosus, which is the third modification of the fetal circulation. The ductus arteriosus also closes around the time of birth. Finally, deoxygenated blood returns to the placenta via the umbilical artery. Notice how the placenta is very much like the lungs in that deoxygenated blood enters the placenta and oxygenated blood leaves the placenta.